Nation, thank you so much for watching. Make sure you like, subscribe, comment, share. It really means a lot to me. Look, really quick video I want to make on stopping criteria for a study or de-escalating. Occasionally in trials, I don't want to say most of the time. I mean, there's always a stopping criteria in any study. It's if the PI thinks that it's causing harm to the patient, or if the PI or if the patient wants to quit, obviously they can stop. But like certain studies have sneaky and multiple stopping criteria, and it's important to know beforehand what those are, and it's important to know what the adverse events of special interest are as well, because like I said, sometimes they're sneaky. So we had a situation today. Shout out to Dr. Smith, our PI. He's the one who saw the lab result. Go figure, he did his job as a PI, saw the lab result, said, hey, this is possibly pancreatitis. We need to look into this. What does the protocol say we have to do? Then we went to check. Command F on my keyboard, put pancreatitis. It showed me 20 things that we need to look at, and 3 to 6 in particular that apply to this particular patient that we're going to do. So it made me pause, because if the PI didn't notice this, if the PI is just going through the motions, didn't say, hey, what's the stopping criteria, coordinators get busy, maybe they miss it, right? So clinical research is a team sport. You need the PI to watch out for labs, ECGs, patient safety. You need the coordinators to kind of know what to do. When I hit Command F, that's kind of like, the protocol walks you through what you need to do, and then the coordinator coordinates with the patient. Hey, let's get this scheduled. Let's come in for another blood draw to run the safety test. The PI gives the clinical opinion. Is it a new diagnosis? What is the current diagnosis? Is it an AE? So understand the stopping criteria. Having cheat sheets. I have cheat sheets for our PIs of different protocols, labs, what we need, what lab results mean, what's next, right? What steps next based on lab results. Not always stopping criteria. Sometimes it's monitoring again, or another draw before you do the stopping or the pausing of the investigational product. It's usually a couple other things, like another blood draw, an ultrasound, an MRI. It could be a lot of things. Then check with the protocols, but most protocols, or some protocols, have stopping criteria, specifically when it's adverse events of special interest. So definitely one of the things you've got to know going in before you do any study is what are the adverse events of special interest. And obviously you're going to know the names, like pancreatitis, but pancreatitis can mean lipase more than certain times above the upper limit of normal. And it might be another enzyme to look at as well. Actually, it's not coming in my head, but lipase was the one that was coming to my head based on our particular situation. So if my PI didn't say pancreatitis, and we just saw lipase 3 times the upper limit of normal, you can ask the PI, well, what is that? What's the medical term for this, or what do you suspect? They will tell you, because they're the doctor. That's why you can never replace a PI or a sub-I, because they're on the trial for their clinical judgment, to be able to give clinical interpretations to situations that you're dealing with at your site. So have everyone on the same page, have everyone know their roles, give cheat sheets, make cheat sheets if you need to, for all the AEs of special interest, particularly when it comes to, if this lab result, then what do you do? And then if this one, then what? There's a lot of that kind of stuff in internal medicine trials, and then there wasn't too many of those in psychiatric trials. So this is something relatively new to me, although I've been doing non-psych trials for 8 years now, maybe longer, but never a day where you don't learn something new. Like, subscribe, comment, share. Understand adverse events of special interest. Understand stopping criteria for studies. And understand how the PIs should be functioning, or sub-Is, amongst your staff. Like, subscribe, comment, share. Bye-bye.